Welcome everyone to Science on Tap. Thank you all for coming out tonight. Science on Tap is a public education science community outreach. We combine curiosity in the community to have conversations. We are so excited to have the EPA back. People have been asking about y'all, so give the EPA a big round of applause. Big shout out to our partners, Radio Artifact and Urban Artifact. They help us put on this event, and Radio Artifact live streams all our events. So if you're super excited about science, you want to see more of our programs, go out to Radio Artifact's YouTube channel and you can see a playlist. With that, I'm going to bring up our speaker to talk about spit and pathogens, Dr. Shannon Griffin.
Come on. All right, thanks, Jen. Okay, let's get started. Got to bring that beer up here, right? Well, I also want to thank Jen and Science on Tap, and I also want to thank Radio Artifact and Urban Artifact for hosting us tonight. So I'm Shannon Griffin, and I'm a biologist with EPA's Office of Research and Development, and I'm going to tell you a little bit more about that in just a second. But I'm excited to tell you about how we use our spit test to understand environmental health risks from all sorts of exposures in the environment. We're going to focus primarily tonight on water-related exposures and specifically on pathogens or those germs in our water that can make us sick. Sorry, words are hard. I swear this is my first beer. Okay, so before we get there, let's talk about where we're headed tonight. So I'm going to give you an introduction to EPA's Office of Research and Development. Then I'll give you a brief overview of federal environmental research in Cincinnati. I'll share some background on water and microbes. Then I'll outline our saliva test development and validation efforts. And then we'll wrap things up with an example application in a drinking water study in Puerto Rico. And so I'm going to draw your attention to the photo at the top. This is actually one of the study sites for that study in Puerto Rico. This is a community-managed water system in the community of Apiadero. And what you see there is one of the residents and volunteer operators. He's working on the electrical panel there, trying to restore power back to that system. This photo was taken in 2023, and he's restoring power to that system because it was lost when a tree fell on it during Hurricane Maria in 2017. So just trying to paint the picture here that there's a lot of environmental challenges that these folks are dealing with in these communities that we're helping to address. Okay, so allow me to introduce you to EPA's Office of Research and Development, or ORD. We are the science arm of the agency, and we provide the scientific foundation for EPA to execute its mandate to protect human health and the environment. And we do this in a number of different ways. So we produce innovative and anticipatory research addressing a whole range of EPA program and regional needs to help identify and solve those longer-term environmental challenges. We also deliver research to support the agency's priorities and help assist our states, tribes, and communities with addressing their environmental challenges. And then we provide technical support to respond to a number of environmental crises and needs, both large and small. And we do all of this, and then some, with an incredibly hardworking and talented workforce, despite what you might be hearing. I am so lucky to work alongside the best and the brightest at EPA ORD. So to all of my EPA peeps out there, and there's a lot of them here to represent, thank you guys for the work that you do. You are the protectors, and we appreciate it. So the other thing that I want to mention about ORD is that we have a presence in 11 locations across the whole United States. We, of course, are here in Cincinnati, and this is home to the second largest EPA research facility in the nation. All right. So now I'm going to give you an overview of federal environmental research in Cincinnati, a little history lesson. So we have a very long-standing history here in Cincinnati of federal environmental research. It started in 1913 when the U.S. Public Health Service set up the Ohio River Stream Pollution Investigation Station. This is the first of its kind in the whole nation. And this took place in the Kilgore Mansion or Marine Hospital. You can see that picture in the bottom left there. Sadly, that's been since demolished. But in 1954, the water pollution research efforts moved to the Robert A. Taft Sanitary Engineering Center. That's the photo that you see in the middle there. This one still exists. You guys probably have all seen this one before. You can see it from Columbia Parkway. That is present day home to our NIOSH colleagues. That's the National Institute occupational safety and health. But what's cool about the Taft building is that the bulk of the federal government's environmental research started there, including when EPA began in 1970, thanks to President Nixon. But then in 1975, EPA moved to its present-day location in Clifton, and that's the building we're in now, near the Uptown campus of UC. So the city of Cincinnati donated a 22-acre site to the federal government, and on that property, we constructed the National Environmental Research Center, which was dedicated by President Ford then in 1975. But then in 1980, we changed our name to the Andrew W. Breidenbach Environmental Research Center, or ABERC, as all the cool kids call it. And that is the building that you see on the far right, and that's where we are presently located. That's actually the building I work in as well. Okay, so I told you a little bit about the history of federal environmental research in Cincinnati, and now I want to tell you about some of the present day activities we've got going on in our building. So we are internationally recognized for all the great work that we do in water research, bioremediation, pollution prevention, homeland security, and emergency response, and that's just to name a few of the things. That's a very broad overview. All of this work is conducted at 4 locations across Cincinnati, and I'm going to tell you about those in just a second. But we cover more than 630,000 square feet of labs, offices, and field facilities. And we're home to approximately 800 employees. That includes 500 federal employees and 300 non-federal staff. The other really cool thing about what we've got going on in EPA Cincinnati is that the research conducted here has broad impacts at local, regional, and national levels, and I would also include global. A lot of my colleagues are world-renowned in their fields, so pretty impressive stuff. Now I want to bring your attention to the photos on the right, and I'm just going to give you a heads up. I'm terrible with left and right, so if I get it wrong, ignore me, okay? But looking at that top circle on the left is the ABERC building, and I already told you a little bit about that. That's where I work. Now, moving to the right, clockwise, you can see the experimental streams facility. So this is an incredibly cool research asset that we have in Cincinnati. It has a lot of artificial stream mesocosms, so you can do all of this neat dosing work and water quality monitoring. And then just below that, you can see some simulated landfill columns. This is at the Center Hill Research Facility. So here we do a lot of work on contaminated sites and waste management. And then following along to the left of that, you see components of the Test and Evaluation, or T&E facility. And this is co-located with the Metropolitan Sewer District Treatment Plant at Guest Street. So what's neat about this is you can do bench, pilot, and full-scale research on water treatment, distribution, and plumbing. So we're really, really lucky here in Cincinnati to have such an incredible group of people doing some incredible work with some really neat research assets. Okay. Finally get into some background. We're getting closer to my work, I promise. But I'm going to talk to you a little bit about water systems, challenges, and health. Okay, so I hope we can all agree that over the last 100 years, many of the improvements in the health, success, and lifespan of the U.S. population can be linked to improvements in water quality. And providing safe drinking water was one of the most important public health achievements of the 20th century. So we provide that safe drinking water a number of ways. Water treatment being one of them, which has helped ensure access to healthy and safe water for millions of Americans. But another way that we guarantee safe drinking water is through government regulation. So that's things like developing those drinking water standards, protecting our source waters, both our surface and our groundwater, and reducing pollution. All of these things have helped ensure the quality of Americans' drinking water. So again, I'm going to draw your attention to the photos on the right. You can ignore the glass of water at the top. That was just because it looked pretty. But the other two are of the Washington Aqueduct. And really, I just add this here to show you what incredible achievements we've made over the years in water treatment and distribution. It's pretty impressive. Okay, that's all great, right? But water challenges still persist. And that's because most drinking water systems in the United States are more than 50 years old. And in fact, most of those are more than 100 years old. So things like cracked pipes, water main breaks, other age-related issues increase the chance for pathogens or chemicals to get into our water, and that can lead to drinking water advisories, or more importantly, to negative health effects. So I'm going to point you to the picture at the top. That is a water main break. This is probably familiar to all of you. We've dealt with this before, but that's one of 240,000 water main breaks that occur annually in the U.S. Yes, right? And that leads to $2.6 billion in losses of millions of trillions of gallons of treated water in the U.S. So major problem. Another issue with aging infrastructure are lead service lines, which you see in the bottom photo. So at one point in time in history, we used a lot of pipes with lead in them as part of our distribution system. Those pipes were banned in 1986, but it's still a problem today. So there is no safe level of lead exposure, okay? But we do have lead service lines in place in our distribution systems in the United States. So it's a real public health problem, particularly for those vulnerable populations like pregnant women and young children. And it's really important that we identify and we get rid of and replace those lead service lines with safer alternatives. Okay. So in addition to all of those aging infrastructure problems, we still have emerging challenges of stronger and more frequent storms, droughts, floods, wildfires. All of these things are heightening the urgency to reinvest in our nation's water sector infrastructure. Okay, so one of the things that I mentioned to you that is a problem is the potential for pathogens to get into our water, right? And the reason why that's a problem is because it can cause waterborne illness. So waterborne diseases, and I'm talking about outbreaks and all of the sporadic infections, they can occur when water treatment or infrastructure systems fail or when untreated water is consumed. Now, most of the time what happens is we get acute gastrointestinal illness, a little diarrhea and vomiting, pretty miserable, but not that big of a deal unless you're a vulnerable person or susceptible where, I mean, in some cases you can actually die from dehydration from diarrhea. But in addition to acute gastrointestinal illness, you can get earaches, skin infections, certain types of pneumonia, even brain infections from water. And these can be caused by more than 50 pathogens of potential concern. That's according to the World Health Organization. But some estimates might be up to 500 pathogens of concern. Regardless of that number, they come in all sorts of shapes and sizes and types, from viruses to bacteria to protozoa to even some worms. So now I'm going to point you to the graphic on the right, and I'm going to talk to you about some of my favorite pathogens. So in the box on the left, I know it's weird to say that I like pathogens, right? But in the box on the left, we have the noroviruses. These are tiny little RNA viruses. They're only about 20 to 40 nanometers in diameter. They are a very large and diverse and highly infectious group of viruses. These guys cause acute gastroenteritis, and in fact are the most common cause worldwide. So lots and lots of diarrhea and vomiting. I guess the one good thing is that it's fairly short-lived, but I would argue it is not short-lived enough. Because if you've had a norovirus infection, you would probably agree with me. It's pretty miserable. So then when we move to the right, we have Helicobacter pylori, or H. pylori. This is a bacterium that's about 3 to 5 microns in length. This guy causes a stomach infection, and it is the leading cause of ulcers and actually can cause certain types of stomach cancers as well. And then continuing on to the right, we have really what is my favorite pathogen, and that's Giardia duodenalis. Giardia is a protozoan parasite. It's a bit larger at 10 microns. This guy also causes diarrhea, and you've probably heard it referred to as backpacker's disease or beaver fever, and that's because hikers and their doggies get it when they're out and about in the woods and they consume untreated surface water. Now, the reason why I like it is because it's the cutest parasite, which you can see, I gave it the award. And that's because when you look at it under the microscope, it looks like a little smiling face looking back at you. That's actually not a face at all. It's just like basically Giardia's storage shed. But regardless, it's pretty cute. The other thing that I want to mention is I put that scale at the top above those pictures just to give you an idea of reference around size. I do want to point out that this is log scale. So Each of those vertical dashes, it's like a multiplication of 10. So the way that you can think of log scale, at least in base 10, is instead of counting 1, 2, 3, 4, we're counting 1, 10, 100, 1,000, and so on. So looking at that, Helicobacter pylori is 100 times larger than noroviruses. But regardless of the size of all of these critters, they are tiny, relatively speaking, but boy howdy can they wreak some havoc on a human, right? And if you've had any of these infections, you would probably agree. The other thing that I will say is all of them are smaller than even the diameter of a single human hair. Okay, so it's really important to know when humans are getting sick from microbes in their water, right? And that's part of our mandate. It's our battle cry. Remember the EPA mandate to protect human health and the environment. So to do that and to better understand the linkages between public health and water, there is a need for tools to estimate health burden associated with waterborne illness. So are people getting sick from their water? Who's getting sick? Where are they? What are they getting sick with? What can we do about it? These are all important questions for us at EPA. So how do we address those questions? Well, that's where our waterborne illness research comes in. And there are lots of strategies for studying waterborne disease in humans, and that can include things like health diaries. So this is one way that we can learn about waterborne disease. The way that this works is that I might give you a form or a booklet or a questionnaire, a little checklist, and you can tell me about what kind of symptoms that you're experiencing. And this is a fine approach. But there's some bias there because I might be asking you about your symptoms in the past day, the past week, the past 2 weeks, the past month. So the reason why we say there might be bias there is called recall bias. Do you remember, did you have diarrhea a month ago? I don't know. You might define diarrhea differently than I define diarrhea. You might be embarrassed to tell me you've got diarrhea. Or maybe you just don't care. That happens too, more than you'd think. Then the other way that we can learn about waterborne disease in humans is to actually look for those pathogens in stool samples. Again, another fine strategy, one that we use. In fact, I tried to prove it to you. And so the photo at the top is a rack of poop samples from our lab. So yes, we do look for pathogens in stool. It works just fine. But aside from it being gross, it's just like people don't want to give up their stool samples. So it's another hard way to get samples from a lot of people. So then there's still another way that we can look for waterborne disease, and that's to look for indicators of infection or antibodies. And you guys have probably heard a lot of about antibodies over the years, especially since COVID. But these are just tiny proteins that are produced as part of the immune response to help us recognize and neutralize pathogens and also to protect against future infections. So most of the time we look for antibodies in blood. Great. Again, fine approach, one that we use, but hard because people don't love a needle stick. And I think the little girl in the photo in the middle would agree that a needle stick is no fun. So that's another hard thing to get from a lot of people, and it's expensive. You need trained phlebotomists to collect those samples. You have all of these special storage and transport conditions, so it makes doing big studies like we do very difficult. So the other place that we can look for them is in our saliva. And saliva is simple and painless to collect even from young children. And I got to give a shout out to my niece Dylan here in the middle. Dylan is doing an excellent job of showing us how easy it is to collect a saliva sample. So thank you, Dylan. She did that as part of a study we did in COVID, and I'll tell you a little bit more about that. So regardless of what we're doing here, it's cool to look at antibodies because in a prospective study, that's one where we follow people over time, immunoconversions, which are characterized by a steep increase in antibody responses followed by a gradual decline, they can be used as a marker of an incident or new infection. And that's really wonderful when it comes to doing these very large population-based surveys where we're trying to collect samples and understand antibody responses from lots of people all scattered throughout space and time. So, taking all of these things into account, EPA ORD developed a salivary immunoassay. Again, words are hard. And that's just a fancy way of saying spit test for antibodies. Okay, so now I'm going to tell you a little bit about our actual method. Okay, so here it is. The Luminex salivary immunoassay, and I'm going to walk you through this. the first thing that we need is saliva, right? So we use the Oracol oral fluid collection device. It's essentially a sponge on a stick. It's right here. Behold, right? Very cool. I wish I had something cooler to show you. I know some of my other colleagues have brought beer and brought their cool carts in, and all I have is a sponge on a stick. But anyway, it's easy to use. And the way that you do it is you rub the sponge along your teeth and gums for about a minute. But what's important about this is it collects what's called crevicular fluid. And crevicular fluid is that stuff that flows along your gum line. And why we want to get that is because it's enriched in your blood components. So it's effectively increasing the number of antibodies present in our sample. And it's getting us a little bit closer to what we might see in a blood sample. So then the next thing that we have to do is process that sample through heat treatment and centrifugation. We got to clean these things up a little bit. We've got all sorts of gook and things in our mouth that get in the way of our assay. But then once we do that, we are ready for analysis. When it comes to analysis, we use Luminex technology. And the one thing that I want you to remember about Luminex technology is that it allows for multiplexing. This means that we can measure antibody responses to a whole suite of different pathogens all at once. That is simultaneously in the single well of a microplate. And how that works is that we start off with these tiny, tiny little beads. They're only about 6.5 microns in diameter, so even smaller than that Giardia that I told you about earlier. But inside of these beads is this unique combination of red and infrared dyes. So it gives it a sort of signature or spectral address. It's kind of a thumbprint for that. But Luminex makes a whole bunch of these different color-coded sets, all with their little internal spectral address, unique combination of dyes inside. So we have all of these color-coded sets of beads to choose from, and to each of these different sets of beads, we can couple proteins, or more appropriately in this case, antigens, from our favorite pathogens. So in looking at the schematic, and I hope you guys can see this, but we would take, say, the blue beads. And to the blue beads, we might couple antigens from the noroviruses. And to the green beads, we'll couple antigens from Giardia. And the orange beads, antigens from Helicobacter pylori, and so on. And then we take all of these different color-coded sets of beads that are coupled to their antigens, and we mix them together in the single well of a microplate. And this is a microplate. And one little circle is a single well. So it's pretty small. I didn't want you to think it was something enormous based on the picture, the size of a centrifuge, for example. But we mix all of those beads together, and then we take our saliva sample. And if there are antibodies present in the saliva sample against any of the pathogens in our multiplex mixture, they will bind those antigens. So then the next thing that we do is we add a detection antibody. And all that does is just binds the salivary antibody that's on the bead surface. And then we add something called a reporter. That's just a fluorescent tag, a signal. Basically, we need that there so when we put this little microplate in the machine, it can see the reporter, what's shining on the outside of the bead. So that's really what the next step is. Take this guy, stick it in the Luminex device, and what it's going to do is it's going to measure two signals. One of them is going to classify that internal combination of dyes. What's the color-coded set of beads that you have present? And then the other one's going to measure how much of that reporter signal is on the outside of the bead. So it expresses that. That output is called median fluorescence intensity, or MFI, and I'm going to use that word a couple of times in the talk. But basically, it's an indirect measure of the number of antibodies present in your saliva sample. So if you've had a recent infection or an ongoing infection with a pathogen, your body's mounted those antibodies, I'm going to see it in your spit. It's going to give me an increased signal, increased MFI when I look at it with this test. Okay, now I want to tell you a little bit about some of the pathogens that we include in our assay. I'm not going to read this full list to you, but the moral of the story is it's a lot. So a lot of bacteria, viruses, protozoa. We don't have any worms yet, but I'd like to. I know, again, weird, right? So in the leftmost column, we have those are the working assays. That means I have antigens from one of my favorite pathogens. It's coupled to the bead surface, and I've been able to validate that it works, that I can actually detect antibodies in a sample from a human that's had a confirmed infection. So that's validated and working. And I'm going to show you what that looks like in a second. Then in the middle column, we have those beads that are, or those assays that are in process. So we probably have a protein coupled. Maybe we just don't have a human-derived sample to confirm that it's actually working. And then in the last column, I have some controls, and we include those to help us understand if there's any kind of non-specific binding or cross-reactivity. Just tells us a little bit more about the background. And then I have a wish list. So if anybody out there, studies cyclospora or microsporidia, for example, let me know. I'd love to talk to you. Okay, so this is our assay in action. Remember that immunoconversion that I talked to you about earlier, where we see that steep increase in antibody responses following infection, followed by a gradual decline. Well, that's this. So when we were developing our assay, we were collecting saliva samples from our friends and colleagues at EPA, and we had all of the proper and ethical approvals in place. I promise it was legal. But we happened to, in the process of this, catch a norovirus infection in the wild. So one of our unlucky EPA volunteers happened to get infected with norovirus while we were developing these tests, and that's what you see here. So I want to get you oriented to the figure. So on the x-axis, that's the horizontal axis, we have the days before and after symptom onset. So at day 0, this person reported having diarrhea and vomiting consistent with a norovirus infection. And then on the y-axis, that's the vertical axis, we have that pathogen-specific response. So again, the MFI coming off of the machine for noroviruses in this case, divided by that control response. Again, an MFI, but really all this is signal-to-noise ratio, okay? So getting rid of some of that background. So we're cruising along, and then on day 0, this person says, oh no, I have diarrhea and vomiting. And what we see is their antibody responses to the noroviruses comes up really high and then starts to tail off over time. But the other thing that you see is we did not see that with any of the other pathogens that were in the multiplex mix. So I've included Cryptosporidium parvum or C. parvum here for demonstration purposes, but basically that just stayed very flat, right? As did all of the other pathogen targets in our multiplex mix. The other thing you might be wondering, why are there 2 lines, Shannon, and what does IgG and IgA mean? Well, the other part of the story I didn't tell you is that we actually measure 2 different types of antibody isotypes. So humans have 5 isotypes of antibodies, just very broadly, and we're looking at 2 of them. IG stands for immunoglobulin, and we're looking at IgG. This is the most abundant in blood. This is the guy that protects you, gives you the longest term protection against pathogens. But we also look at your IgA response, and that's found in mucosal secretions like tears and saliva. This one provides protection in those areas, but also your respiratory and intestinal tracts. All right, so that's an immunoconversion. This is like what we're always looking for, and we happen to capture one. Now I want to talk to you about validation of the assay. And in this case, we're validating the assay using samples from a norovirus human challenge study. So in this case, people were fed noroviruses. We did not do this at EPA. This was done at Emory University. We were collaborators, and they did give us some samples. But the way this worked is that they actually took oysters, and they used high hydrostatic pressure, and they seeded noroviruses into these oysters. So you had a group of oysters with viruses in them, and you had a group of oysters that did not have viruses in them. And again, every line on this graph is an individual. This is a subset of individuals from this challenge study, so there's only 7 here. Each line representing one of those individuals. But let's take a look at how to read this figure. So the X horizontal axis, we have the days post the norovirus challenge. This is day 0 was the oyster feast, right? And then on the Y or the vertical axis, what we have is that signal to noise ratio like you saw before, but what we did here is that we looked at the signal to noise ratio at any given point in time and expressed it as a ratio to that baseline day 0 oyster challenge response. So all we're doing is looking at the fold increase in the antibody response over time. And the reason that we do that is because in the scientific community and in the literature, we define an immunoconversion or seroconversion, as we call it in blood, as a 4-fold increase in a specific antibody response over time. So that's the big red line, the horizontal line across. That's the threshold that you got to beat to have an immunoconversion. So what we saw is that the 3 individuals that ate we'll call them the crappy oysters, they were infected, and they had symptoms consistent with a norovirus infection, and they also shed the virus in their stool, which happens when you have norovirus. But the 4 individuals who ate the plain old oysters were not infected, and they did not have symptoms, and they did not shed the norovirus in their stool. So again, this is just one way that we can validate our assay. We've done something similar with Cryptosporidium. We have a collaboration with a global pharmaceutical company that we've been working on with a much larger sample size. We're seeing some similar things, but that work's not published, so I'm not going to share it tonight. Okay, so finally, let's talk about how we use this method to study environmental exposures. Beginning in 2006, that's actually when I started with EPA, and we started developing these saliva assays. Since that time, we have collected and tested over 50,000 saliva samples for evidence of infection from a whole range of populations and locations, including marine and freshwater beach sites. So with this is a recreational water study. So for these studies, what we were looking at were beaches that were potentially impacted by sewage discharges, treated sewage discharges, but from nearby wastewater treatment plants. And we wanted to see if there was any swimming-associated illnesses in these people. So that's one way we used our method. We've also done a number of population-based health assessments in Colorado and North Carolina and Massachusetts, and even in Puerto Rico. And then another thing that we've done is partnered with a number of tribal nations in the southwestern U.S. looking at their environmental exposures. Again, primarily that's around drinking water. And then the last thing that I want to call out is that during the COVID-19 pandemic, we pivoted our time and resources to develop a saliva assay for SARS-CoV-2. And remember, SARS-CoV-2 is the causative agent of COVID-19. What was really neat about that assay was 2 things: that we could discriminate between vaccine-induced immunity and naturally acquired immunity. And then the other thing, that whole study was done by mail. So people were literally just collecting their saliva samples safely at home. They're not putting anyone else at risk. There's no healthcare provider there to collect the samples, so you're limiting exposure to the people around you. And they would send their samples by mail back to us in our EPA labs for analysis. So this was all part of EPA's emergency response research, and was another really cool project. Now I want to do a deep dive on one specific application, and this is the study in Puerto Rico that I talked about. This is a collaboration with Inter-American University's Center for Environmental Education, Conservation, and Research, or CESIA in Spanish. We had 2 objectives for this study. The first one was to investigate the health impacts associated with consuming inadequately treated drinking water from community-managed water systems in these remote, disadvantaged areas of the island. And then the other one was to evaluate the public health benefits of installing lower-cost, sustainable filtration technologies in these underserved communities. So you can see some members of the study team there in the top circle, and then as we move clockwise, you can see we're out in the field. I think we were troubleshooting a problem with the source there at the community of Apiadero. That was the first system that I showed you. And then in the bottom photo, you can see the system that was installed in the community of El Real. So these are some pretty impressive structures that have been built as part of this larger effort. And we'll talk more about those. So let's better define the problem for this project. So we know waterborne pathogens present public health and economic challenges in Puerto Rico, and in fact in the U.S., the contiguous U.S., I will say. But 240 drinking water systems for domestic use are not connected to the Puerto Rico Aqueduct and Sewer Authority. These are the so-called non-process systems. They're not receiving municipal water. A lot of times these are just community managed. And because of the rough terrain and remote location, it makes it really, really difficult and expensive to provide service to these disadvantaged communities. And what's more is that they have a historical high percentage of health-based violations for their water quality. So this represents a risk to public health. And these compliance problems come as a result of economic limitations, a lack of adequate treatment, poor sanitary conditions, a lack of maintenance programs, and even a lack of qualified operators. So taking all of these things into account, you can imagine that these systems are very vulnerable to natural disasters, climate change, and other environmental challenges. So I want to draw your attention to the photo on the right. What you see here is just some PVC pipes running down the hillside from a surface water source through the woods. This is being piped directly into people's home with no treatment, no disinfection. This is not an uncommon sight in these communities at all. So let's talk a little bit about the drinking water intervention study and the study design. But first, just to orient us, in that top map, in that purple box, I hope you can see it, that's the island of Puerto Rico. And you can see it's a ways out there in the Caribbean Sea. So you can see the tip of the Florida Peninsula and Miami there in the top left. So Puerto Rico is out there in the purple box. That's my mom. It's cool. All right, it's all right, Mom. So if we zoom in on the purple box looking at Puerto Rico, that's the map in the bottom. And in the orange circle, you can kind of see that. Yep, that is our study area. And I want you to kind of remember about where that is because we'll revisit that later on. But basically, the plan for this study was to enroll 7 small communities near Patillas, Puerto Rico. That's within the orange circle. All of these are non-PRASA communities. This is an intervention study, so we're going to install those low-cost, sustainable treatment technologies in 3 of the communities. So 3 intervention communities, 2 of them are served drinking water treated by biosand filter system and chlorination. These are the communities of Mulasagual and El Real. And then 1 would be served drinking water from a modular conventional treatment plant using filtration and chlorination. That's the community of Apiadero, which you saw at the beginning. And then we would have 4 control communities that would have no enhancement of their current treatment, or lack of in most of those cases. Okay, and we'll do a deeper dive on the study design. This study has 3 phases. The pre-intervention phase, this is where we would administer baseline survey data. We're collecting all of that information about your basic demographics, your household size, your household income, education level, things like that. But we would also collect information on health diaries. We want to know about the symptoms that you're experiencing. And there is this separate water quality monitoring part that's happening throughout the pre-intervention phase. During the pre-intervention phase, EPA wanted to do a pilot study. And what we did was collect cross-sectional saliva and stool samples. That's just one time from a subset of participants in the bigger study. The reason why we did this is we wanted to see when you go to people in Puerto Rico and ask them for their spit and their poop, are they going to give it to you? What do they think about that? Can we get these samples? And this was all in an effort to inform the post-intervention phase of the study. So the second phase of the study is the construction phase. That's where we identify, design, and implement all of those treatment technologies in our intervention communities. And then the very last and final phase is the post-intervention. And here we're going to have a prospective study design. It's actually expected to begin this summer, perhaps even this month, fingers crossed. But I do want to tell you a little bit more about what that prospective design looks like. So again, start with those 7 communities. We have our 3 intervention communities in blue. And we have our 4 control communities in green. Regardless of which arm you are in the study, the sampling strategy is the same. So at baseline, we're collecting our survey data, all those baseline demographics. We're getting our health diary. We're collecting a saliva sample and a stool sample. And then we're going to collect a saliva sample and health diary data for 3 months at monthly intervals until the end of that 3-month follow-up where we'll collect an additional stool sample. If someone reports gastrointestinal illness or symptoms consistent with that, throughout that 3-month follow-up, we will ask them for another stool sample. And of course, we're going to continue with all of that water quality monitoring as well. Okay, so where are we with this study? Well, the pre-intervention and construction phases are complete. And thanks to an incredible team at SACIA, they were able to enroll 821 residents. They've got tons of data on all of those household surveys and health diaries and water quality and all that good stuff. But as for the EPA pilot effort, we did get 402 saliva samples from 174 participants and 170 stool samples from 141 participants. And this study has been met with so many challenges. It is insane. We've had numerous hurricanes. We've had earthquake swarms. We had a pandemic. So there's lots and lots of delays, but thanks to the persistence and the stick-to-itness of incredible community partners and collaborators at SACIA, and I gotta say, toot toot, EPA, I'm gonna toot our horn too, but we really worked hard to try and make progress on this study, and we've succeeded thus far. So I want to, again, draw your attention to the top photo. That is the construction of the biosand filter unit at El Real, yep. And then below that, you can see on the right-hand side, you can see where the tree kind of fell on that system in Apiadero and knocked out power. The other thing, maybe, I hope you guys can see this. Maybe not. It's a big tarantula on the pipe. So I just put that there to highlight some of the other challenges that we deal with in these communities, and it was huge, but it's kind of in the shadow of the why. All right. So just a quick summary of our pilot. As for the saliva analysis, we did observe strong associations between gastrointestinal symptoms and increased antibody responses to noroviruses. So good for us, we could see it. Bad for them, they're getting sick. The other thing with the stool analysis that we saw was 15 individuals, so that's almost 11% of the population that we tested, was positive for some kind of pathogen in their stool. A lot of usual suspects here, things like Shigella, Cryptosporidium, Giardia, noroviruses, those were all present. But the other thing that was a little bit concerning, at least to me, is that we saw Entamoeba histolytica and enterotoxigenic E. coli, or ETEC. These actually in children at that, which is sad. But these guys are sometimes indicators of poor sanitary conditions. So you can imagine if you have poor sanitary conditions and you're not treating your drinking water, you're really setting yourself up for a major public health problem, right? So the other thing that I want to say about the pilot phase of the study is that, remember, the point of that was to inform the post-intervention. So I think it's important to capture some of those lessons learned. It did serve its purpose. We did gain new insights regarding an optimal strategy for sampling in the post-intervention. But the other thing that we learned a lot about was working across barriers. And I would say language was the least of them. Trying to figure out how to get all of this equipment and these tools up these mountains into these remote areas was incredibly difficult. We also did this without cellular service or internet. So you couldn't even troubleshoot when you're up at the site. You'd get up there and realize there's a problem. You have to hike back down, drive 45 minutes down into the city, hope you could get some service, Google it, go to the hardware store, go back up the mountain. So it is quite the challenge working in some of these communities. But the way that we were successful is because we had, again, incredible community partners. And that is so vital to the success of this study, and I would argue any study. So that being said, it's important to avoid community fatigue. People get tired of hearing from us. They get tired of responding to us. I am tired of listening to myself right now. So I can imagine, working in these communities becomes problematic. And then the last and maybe most important lesson, I think, is that we should emphasize lived experience as critical data. We as researchers and scientists have a tendency to want to just observe the world and measure the world around us and crunch those numbers and analyze it. And when you're working with people and in communities, sometimes you forget that you really need to think about their experiences. They know their problems long before you do as a scientist. So it's really, really important to listen to the people that we're serving. And so I think this is something that I'm seeing a lot more movement in the science and research field, which is heartening. And as for the pictures, so the top photo is one of the outreach and engagement events that our partners at SACIA did. So they hit the schools pretty hard, which was really neat because the kiddos were getting excited about public health and water quality, and they'd take that home, and then they'd encourage their parents. And so we got a lot of engagement and recruitment in the studies just working through the schools. And then the bottom photo is to show you that this is the chlorinator and water storage tank of the system at El Real. And the community worked with some volunteers to paint a mural on that, which I think is neat because it shows some buy-in and some excitement about the treatment technologies. Okay, next steps. We're almost done, I promise. So we have the post-intervention phase I mentioned to begin this summer. Once we have all of these data, we're going to be able to compare the rates of waterborne illness in our intervention and control communities. But most importantly, we're going to get at that big question. we're going to be able to look for epidemiological associations between water quality and health effects. The other thing that I hope is going to come of this study, well, first, we know that this is, or actually to our knowledge, it's the first comprehensive assessment of waterborne illness and water quality in these non-PRASA communities. We've also, I think, successfully demonstrated the utility of our non-invasive saliva test in a community where a serological survey would be impractical. And I mean like drawing blood. And I say impractical, I would say impossible for that to happen in these communities. And then the last thing, and one I think that means the most to me at least, is that we were promoting community empowerment, decision-making, and capacity engagement all through this engagement and information sharing. And the point is just to advance communication, understanding, action, and most importantly, solutions for these communities. So again, the top and the bottom photo is just to show you some of those outreach and engagement, and that we're getting a lot of buy-in and folks are participating in the building of these systems. And then the other thing, I just want to show you where the eye of Hurricane Maria was in 2017. And you see that aligns almost perfectly with Patias, the area where our study occurred. So these folks still haven't recovered from Maria in 2017, and we are headed into another hurricane season, perhaps with reduced funding. So hopefully things work out here. All right. So I want to just kind of wrap up with our key takeaways for the day, but I want to take you to the photo at the top. This picture was taken by my dear friend, colleague, and partner in crime, and I don't mean that literally. For this study, it's Paige Jordan, and this is a storage tank for groundwater in the community of El Real. What is probably not clear right off the bat, but if you look a little closer, just feet away from the edge of that tank is an insane landslide. I mean, literally, it just falls off. And so this is a real problem for this incredibly important, vital water source for the community of El Real. The last that I heard about a year ago, the community was working with the Army Corps of Engineers to stabilize that and protect the integrity of that tank. So we'll see where that goes. But I just want to wrap up by saying that we know waterborne illness continues to present public health and economic challenges in the U.S., and that's despite advances in water system infrastructure. The key takeaway number 2 is that we developed a non-invasive saliva test for detection of infections from waterborne pathogens. And then lastly, the saliva test provides improved estimates of waterborne disease, and it supports communities in prioritizing their risks, but also informing decisions around their water quality and treatment that helps improve the health and welfare of not just those communities, but for all American communities. Finally, I want to show you our research team and give some acknowledgments here. Of course, our community members, our study participants, and interviewers. We couldn't do any of that or any of this without them. We've got a lot of great partners at SACIA. We have our contract support from Pegasus and Tri-Hydro, a ton of people at EPA. I even had to give a shout out to the hardware store in Puerto Rico because they saved us so many times. And I learned a lot of great Spanish, trying to troubleshoot these problems there. Okay, so I'm going to close with a photo of the system at Mulas Jagual. This is another one of those biosand filter units. But what you see here is some of the members of our team at EPA working directly with the community members. And the reason why I show this is because it's beautiful, but what I think is really even more beautiful is that these systems are being built by the communities that they serve. So EPA does do a lot in the way to help with design and even providing resources, and we provide labor at times. But there is this real sense of ownership and a real pride in operating and maintaining these incredibly vital water resources. So I want to thank you guys again for coming and listening to me talk for, 45, 50 minutes. I don't even know what it is right now, but I'm happy to take any of your questions. Thank you. Yes.
The people who were drinking the untreated water, that's the water they're normally drink, isn't it? So are they like sick all the time?
That is correct. So people who are not getting the treated water, yes, in fact, sometimes they are getting sick. Sometimes they're not. So the other thing that we know with our immune response, once you encounter a microbe, you get some level of protection for some amount of time. So the other, thank you, you just gave me like, I get to put in a plug, because the other thing that our test does is it can measure asymptomatic infections, which may not be making you sick or you're not having symptoms, but you're still contributing pathogens to the environment for those people who are vulnerable. So it is the case that, yes, they might not, they're still drinking that water and maybe they're not having a lot of symptoms, but they are still contributing to the environment. And the other thing that we're learning is that there are some microbes that can cause other types of sequelae or dangerous, like, Epstein-Barr virus, things like that can cause other types of, we learned about long COVID, right? That you can have other types of symptoms that show up after infection. So it's still important to make sure that we're addressing those challenges from microbes in the environment. Thank you. Yeah, Logan.
Shannon, I'll ask a weird question.
Okay, I'll give you a weird answer.
In the study with Emory where you had the oysters,
Mm-hmm.
they get the norovirus or the control. So you mentioned that you were collecting the stool and looking at the antibody response. Did you collect the vomit too?
No. So Logan asked if we collected the vomitus from the norovirus challenge study at Emory, and the answer to that is no. And I'm glad that we didn't have to test it, because out of all of the matrices that we could test, that's got to be my least favorite. But what they did, so Emory led that effort. They did collect blood, they collected the saliva, and they collected stool samples, but no vomit. Yeah. Yeah, any other questions? Yeah.
What is the difference between the two types of the filtration system, the biosand and the other one we talked about?
Yeah, so the biosand filtration system has just that. It has this biological membrane on the top that does some level of mediation, where the other guy was just like a classic like sand filtration or granular filtration. So the reason why they're a little bit different is the community in Apiadero is tiny. It's like maybe 100 residents. And so the number of households that were on that system initially, I want to say was somewhere around 30, whereas with the others, they needed these much larger units. And so that's why we included the biosand filtration units there. And it just confers another layer of, I guess, treatment, if you will. I don't know who was first, but I'll let you guys duke it out.
So, in like a microbiome, when you're collecting the saliva samples,
Yeah.
what I know from microbiome is there's so many different types of microbes all over the body, much less inside the mouth.
Yeah.
Are you missing something if they don't like hit the roof of the mouth or the underneath the tongue? Something?
Yeah, so that's an excellent question. And the cool part is, so we're not collecting we're probably inadvertently collecting microbes and cells and whatever other crud is in your saliva sample, but we're processing the samples and cleaning them up and just capturing the antibodies. So even if you had, let's say you had some kind of infection in your mouth and we collected a microbe, it's not going to bind the antigen on the bead surface, because those guys don't interact. So it's just going to get washed out in our assay. So really the only thing that we're going to bind is like what's going to bind those antigens, and we know that antibodies bind antigens. And then we use that secondary detection antibody as another layer of to keep it specific to just the antibodies that are in your spit. Yeah.
So can you speak a little bit to how the results will be used? Because presumably having some sort of treatment is better than not. So what happens with your data?
Another great question. And so, sorry, I realized I was supposed to be repeating these questions. So she had asked about how we're going to use these results. So it is true that EPA has an obligation for accountability, right? So when we put these environmental regulations out into the world, we have to show, even if intuitively it makes sense, we have to show that when you comply with environmental regulation, you actually improve public health. So this is one way that we're just demonstrating our obligation to accountability. But the other thing is, if we see that one kind of, filtration system works this way and we see this kind of reduction in illness, then maybe that'll help us inform another system that's having similar problems or other communities. Remember, there are 240 systems in Puerto Rico that are these non-process systems. So we're only looking right now at 3 of them. And I will say that our friends and colleagues at CECIA are working right now to start trying to implement these lower cost filtration technologies in some of those other communities. So these are kind of a demonstration project. Do they work? Another big part that I didn't even get to speak to for this talk was the enormous amount of training that we put the community members through on how to operate and maintain these systems. Because at the end of the day, if they don't use them or they hate it, the wheels fall off, right? Then we know, okay, back to square one, that's not gonna work in another community. So that's another way that we're using these data, is just kind of logistically, is this a good idea? Is this a practice that others can adopt? Yeah, sorry.
About how many samples do you need to get a good idea of what diseases are in a community? So that you know you're not just getting a bunch of people who happen to have the norovirus.
Yeah, so he asked about how many samples we need. So there's 2 parts to that. The first one is, how many samples do we need from a single individual? And the answer is at least 3, because I need to be able to see here. I want to try and catch that, because we're constantly comparing your antibody response to yourself at some other point in time. So generally, I would say at a minimum 3. Other studies that we've done, Katie here and I are working on a study with some folks at Temple University, a groundwater study on kiddos. And we have samples collected at monthly intervals for a year, which is great. So, but that's expensive, and logistically it's difficult. So the minimum number of samples that I want to see from any individual is 3. But to the other question, how many people do I need to sample? Now that's a bigger and more complicated problem. And so for that, we have to learn something about the community. We have to have some prior knowledge of what are the baseline rates in a community. We can, like Mike Jane here in his quantitative microbial risk assessment work, we might get some knowledge from there to figure out what's the prior. How many people do I need to sample from to see a difference in if I put filtration in place or not? So we do things like power analysis. We use statistics to help us define our sample size. So that varies. I don't have a really easy answer there. I'm sorry. You had another question?
Yeah, so going back to they stopped using the system, so you're changing the quality of the water, and it might just taste weird to them because they've been drinking those untreated water for decades.
Yeah.
So is that or they say, well, we're not getting sick anymore, we're cured, let's just go get water out of the well.
Yeah, we actually are having some problems with that, to be honest with you, about just maintaining that level of buy-in. So when we administer our questionnaires, we do ask questions about the taste, the odor. So we want to know how people feel about the quality of their water, which is an important indicator. Because the other thing is, people are like, well, clear enough, drink it up. But they can't see these microbes in there, right? So a lot of it's about education as well, as saying, I know it looks clear and I know it tastes good and I know it smells fine, but there are other things that we have to do to make sure it's actually safe. And that is really our goal at the end of the day, is just making sure that they have access to safe and reliable. That's another problem here in Puerto Rico. They have rainy season, they have a ton of water, they get high turbidity running down the mountains, and we need to clean that up before they drink it, and then they go into their drier season, although I wouldn't necessarily call it dry, right? So then they start storing their water in cisterns and all of these things. So that's another project that folks are working on and at EPA in Puerto Rico, is what do we do with the people who are kind of storing that water away? Are they maintaining it? Are they disinfecting it before they drink it? So there's a lot of complexities to these challenges for sure. Yeah?
Yeah, so how far out is your funding secured? What time is the elephant in the room?
I'm just going to backwards walk out of here. Oh boy. Char, help me. No, it's tough. I will say I don't have funding right now. Okay. And any mechanism that I had, like I'm a purchase card holder for the government. That is, that's been cut off. My limit's down to $1. So I don't have a mechanism right now to buy things. There are some strategies. We have some workarounds, but as of right now, I don't have any money. And some of our contracts have been canceled, but our friends at, Inter-American University and Stacia are continuing to forge ahead. So they're going to move forward with this post-intervention phase of the study, and they're going to collect the samples, and we're just going to try and kick things down the road and hope for the best. So I think that's probably all I should say. Yeah.
So we had a different Science on Tap in September of 2024 that was Dr. James Goodrich. And he brought out a thing called
The wild card, yeah.
And I wanted to see if you had any samples or done anything in conjunction with that.
We have not, but we should. And you know what? I mean, if we got to keep things in-house for a little bit, you just gave me a good idea on how to occupy my time. Thank you. Yeah. Any others? Logan. Oh, it's water on wheels. I actually don't know a lot about it, but it's like this portable device, right? And it's used in emergency response. I'm like looking at all the other EPA folks in the room. But I think it's a way to clean up water in emergency response situations, right? And it's portable and tiny and cool, and it avoids using bottled water. Thank you. Yes, okay. Yes. Oh my gosh, Mom. Nope. Yes. Any others? Oh my gosh. Oh, no, okay. So I won a very cool award, the Arthur S. Fleming Award. And you guys probably saw this email. Yeah, thank you. Back in November, I got to go to DC and receive this award, and it's just for excellence in federal service, and it was for a lot of this work around the development of the saliva test. So that is my mother, you know, she's got to do these things. Yeah. All right, others? Oh, yep, we got some more. Oh, are we good? Okay.
Some of the studies you showed that you use the fluorescence technique
Uh-huh.
to analyze the optical spectrum. Basically, that is the linear response of the sample. But that is not touching the response of the polarity of the sample. So I'm wondering if to get information about the polarity of the sample will help you to distinguish the result better. What are the bacteria that you have? What are the characteristics? Because in that case, the technique that takes into consideration a response to the polarity, it will be very useful.
Yeah, so I think what I heard is that the method that we use looks at the fluorescence, but it misses the polarity. Is that what you're saying? Yes. Okay. So I think that would be very important if we were actually looking at the microbes themselves, at the bacteria, learning about the complexity and polarity there. But our assay is only capturing the antibodies. So I think that would come. That wouldn't come into play as much, and maybe we can talk a little bit more about that so I can better understand your question. But in this case, we're just looking at immunoglobulins, and especially with IgG and IgA. I mean, IgA is a dimer, IgG is a monomer, but otherwise they're very consistent in their structure aside from, you know, those little heavy and light chains. But otherwise, I think we don't have to worry so much about the polarity of that part, but I do agree that if we were trying to capture the actual microbe, then that would open the whole door for lots of other things that we should be considering. Does that help? Okay, yeah.
How many pathogens can you multiply?
So in theory, Luminex claims that they have like 500 bead sets that you can use. No way. Not for what we're doing, okay? If you're doing like maybe nucleic acid stuff where you can actually couple the oligos to it, that might work. But when it comes to proteins with these pathogens, we've been pretty successful, I'd say, with like 30 to 35. Would you agree, Katie? We're gonna try and push it over the next few months. We do have some things left in the lab to play with, so we're gonna see if we can keep building on to that. But in theory, our instrument can handle 100. In practice, I would imagine it's less than 50. But we're at 30. You know, we're doing okay with 30, which is pretty good. Yeah. Okay. Yeah. Last question?
Does the Cincinnati EPA branch do any work with other regional organizations like WAVE or
Orsanco, yes, and lots. Actually, we have so many wonderful people in the room that are doing all of this work in connection with some of the other local organizations. Me personally, I don't know why, but most of my work is like outside of Cincinnati. So I've done a lot in Puerto Rico, I've done a lot in Massachusetts and North Carolina. So I can't speak to that too much, but I can get you in contact with the folks that you might want to talk to if you have some more questions about that. But I know Orsanco was a big one. Any other big ones I should shout out? We'll go with that. Okay. But I mean, we actually have a community engagement coordinator that it's like their job to interact with the local community. So I think that's pretty cool. We do outreach in schools and things like that. So very much so, opportunity for that. All right. Done. Thank you guys again. I appreciate you listening. Thanks for your attention.
Thank you so much. That was fascinating. For those that want to know more about the wild cart, that video is out on YouTube. I will share out the link again because it's super cool. And I do get questions from folks in disaster areas after tornadoes go through, like, how do I get one of these? And I'm like, I don't. I'm not the person, but I try to get them to the local resources in that area. Thank you all so much for coming out to Science on Tap. Again, a big shout out to the EPA. Thank you for protecting our health, our environment now and for future generations. We appreciate you. Science on Tap is the first Wednesday of the month. Next month, we're going to be talking more about water. We're going to talk about the Great Lakes region and how that was formed and how important it is to our part of the planet. So come out for that. September, we're going to be talking about moths. Butterflies get too much attention. We're going to give some love to the moths and learn about how cool they are. And in October, we're going to bring back our Science Shots program. Think of it like TED Talk meets research graduates from UC. We're going to learn a lot of science in a short amount of time, so that's going to be a lot of fun. Again, thank you all for coming out. You can follow Science on Tap on Facebook or on Meetup. And again, thank you to our partners, Radio Artifact.